you. Welcome to our Globe TV's amazing news. The engineers and biologists at Dartmouth College have found that just like word order affects meaning on a sentence, the placement of genes within plasmid known as gene syntax can influence how strongly each gene is expressed, how consistently it behaves across cells, and how it interacts with nearby genes too. DNA sequences of each individual gene remained unchanged, yet altering their syntax resulted in different expressions levels and interference with neighboring genes. Results show that gene syntaxes affect how transcriptional machinery moves along DNA, and how replication and transcription processes interact, and how much variability is introduced into gene expression. A plasmid is a small circular piece of DNA that exists independently of a cell's main chromosome, often found in bacteria. Plasmid can carry genes that are useful for survival and can be engineered in the lab to introduce new genes into cells. Scientists use them as tools to study gene function to build synthetic biological systems for genetic engineering and biotechnology. Viewers, synthetic plasmids often exhibit unpredictable gene expressions behavior that have not been completely understood. Intragenetic elements like promoters and ribosomal binding sites have long been the focus of plasmid optimization, yet unexplained variability remains. Even with identical genetic sequences and backbone components, genes may have differently than predicted, and this has left a lot of biotechnology research stuck in logical guess and check mode, where known elements with understood functions just don't operate as they should. In the study, gene syntaxes, modular gene expressions and circuit behavior on plasmids, published in the Journal of Biological Engineering, researchers conducted a systematic experimental analysis to determine the influence of gene order and orientation on plasmids driven gene expression. Each plasmid construct was designed to isolate the effects of syntax without altering co-regulatory sequences. Plasmids were constructed using E. coli, that means Escherichia coli NAV10 beta cells. Fluorescent reporter genes were arranged in different orders and orientations within the synthetic plasmid, while all promoters and ribosomal binding sites remained identical. Gene expression was tested across 7 distinct plasmid configurations. Flow cytometry was then used to measure gene expression in over 9,000 individual bacterial cells per sample. BN's exclusive report.